Thank you, Professor Abhijit. Thank you, Shubhanjan, for this wonderful opportunity and to be here for this meeting. So essentially, what I will be talking about, this word that I'm going to talk about, mostly. So that is in collaboration with Professor Shilpi Dutt from Actric. Now she is in GNU. Essentially, this collaboration started in one of such gathering, and over the lunch we were discussing something, and then we thought that okay, we can work together. So Shilpi's student Anilha and my main student Pallavi, they started the work together, then taken up by Ketki, and this is the kind of one part of the work. And then Dr. Arpita, who is a postdoc, joined my lab, and she taken the work forward with the present collaborator, Professor Mohit Jolly, and his student Sandhya from IISc. So, but before I go into today's talk, first let me talk about from where I come. So Paris is known as City of Light as well as City of Love. So this is a night view of our campus. It is, again, you can see that also a beautiful with the lights. And the campus is essentially, I come from IIT Bombay, between two lakes and the hills, as you can see here. And this is the westernmost part of the country. And yesterday, with someone we were discussing that from the easternmost part of the India to the westernmost part of the India, time gap is 2 hours. The local time gap is 2 hours. So that's why we say in India we have many countries inside one country. So IIT Bombay, we have 10,000 students and various different I think 34 plus department plus centers. I belong to Department of Chemical Engineering, where we have 45 faculty members working in various different areas of chemical engineering. So what my lab do? So what my lab does, so when I started my lab, we started to call it the M lab. M here stands for, that point of time I thought my lab works for the mechanobiology and the microfluidics, both are with M. I am Majumdar in Mumbai, so it's a nice M lab. Later on, in the last 10 or 11 years of journey, I have realized the M actually has a deeper meaning. And what we are essentially trying to do is to mimic the microenvironment in our lab. So the tissue microenvironment that happens in our body, that we try to mimic in the lab, and that is the bigger picture that we are trying to do. So when I say mimic or mimicry, it is not making a clone. I am not trying to make a clone of something, but I am trying to mimic. So that what is the meaning of mimic? Meaning of the mimic is that you don't need to copy every finite details. You need to realize or recognize which are the important details that if you copy, it will serve the purpose. Like this. You don't need to copy everything to tell what this is or what it is. But it can capture the essence that we want to capture. Similarly, in our lab, we are trying to do is that we are using one arm, that is our mechanobiology arm. There we try to understand how the various different mechanical property of the tissue, like rigidity, structure, porosity, topology, that may influence the various cellular behavior. And on the other hand, we are working with the microfluidics, where we are trying to mimic the perfusion, shear stress. Yesterday during the talks, we heard from Rajdeep's talk about the shear stress. So that is something we are trying to do, and from that we are trying to make various organ on chip. Again, yesterday we heard about the liver on chip and the gut liver on chip. So that is what we our lab is working on. For today's talk, it will be mostly focused on this mechanobiology part, and there we will be particularly focusing on our own application, that is the glioblastoma. So for the general kind of discussion, so that we all are on the same page, what is glioblastoma? So glioblastoma is essentially one of the brain cancer that forms that starts from the glial cells, and this is one of the deadliest cancer. If one person gets detected with glioblastoma today, with all the surgery, chemotherapy, radiotherapy, everything, you can remove the primary cancer of the primary tumor, but almost with most certainty within 6 to 10 months the tumor will relapse, and the relapsed tumor is known to be much more aggressive. And the thing is that this is from 1967, this is from 2014, things have not changed in the last 10 years also. You can see that the life expectancy has not changed much. Even in the last 50 years, we have progressed a lot in many other cancers. In many cancers, we have often say that, okay, we have got the win over this cancer. Not true for the glioblastoma. We have not moved much. And one drug that is available, that mostly used, the chemotherapeutic drug, is the TMZ or temozolomide. Now, as I was telling, is that it is the parent tumor, you do everything, but still you remove the tumor cells, not all the cancer cells that you can remove. Okay, some will still stay back, and then they form the relapsed tumor within 6 to 8 months. And it has been clinically seen that the relapsed tumor is much more aggressive. And when I say that aggressive, the meaning is that they are highly proliferative, highly invasive, they are drug resistant. So in some sense, I can say that if a patient is going through the treatment, then the patients are dying not because of the parent tumor, but because of the relapsed tumor. So we need to really understand the biology of the relapsed tumor, and for that we need a model. Now you need to understand here one more interesting thing, is that, as I said, the parent tumor will go through the surgery. It will, after that the tumor will relapse within 6 to 8 months, but that time it is highly invasive. It has almost covered. It is not coming.
Yeah, because could you share the slide with because people are
sorry. Sorry about that. Don't count the time. Disruption. So as I was telling you, is that we need a relapsed tumor model. But one thing you to understand here is that for the parent tumor, as I said, operation is done, surgery is done, everything is done. Within 6 to 8 months, the tumor comes back. The relapsed tumor is highly invasive. It has spread every part of the brain. So most of the time, the clinicians or surgeon will not go for a second surgery because there is no point. So as a result, getting a paired sample of the parent and the relapsed tumor is very rare. So for that, in Shilpi's lab, what they have developed is that they take the cell line or sometimes even the patient sample, but from the parent tumor, they expose them to the radiation, to the lethal dose, so that most of the cells die. If you look the Petri plate under the microscope, you will not be able to find cells. But if you keep the Petri plate in the incubator, within 7 days, some large giant cells will come up with multinucleated. Then they will get cytokinesis, and there we call it the relapse or the radiorecurrent tumor. And then we check their some of their markers for the relapse, and they say that okay, this is established. This can be used as a model for the recurrent tumor cells. However, as I told you, you remember that clinically relapsed tumors are much more aggressive. They are highly proliferative, highly invasive, drug resistant, hypertensive. When we take this care and we check for, say, migration, invasion, you can see that the recurrent one has a higher value, but not really very statistically significant. So that gives us the question, is this model that we are creating are not really capturing the, the behavior? But then when we put them in the mouse brain, you can see the difference. Clearly the recurrent tumor are proliferating more as well as invading or migrating more compared to the parent cells. So something is happening inside the brain, the microenvironment, which is not happening when we are doing it in the petri plate. That's basically the crux of the question here. So when we were discussing with Shilpi, my collaborator, and then we talked about that brain is essentially the softest tissue. It is like a jelly, and we are culturing these cells on a hard plastic. Are we missing something? Also yesterday, I think Atif discussed that the tumors, they change their rigidity. I think Rajdeep also discussed that. So when in case of the glioblastoma, it is the looking at the tumor grade, initially the stiffness goes up, but then the stiffness goes down. So it looks like stiffness has some role to play. Further, taking U87Mg, which is a cell line, another cell line, patient sample, we compared parent and the relapse cells, looked into the transcriptomic data, did the pathway analysis. Differentially, what comes up at the top are the focal adhesions and ECM receptor interaction. Again, the whole thing tells us that the difference between the relapse cells and the parent cells are lying how they interact with their microenvironment. So with that, we ask the question, what will happen if can I create the recreate and can I capture this aggressive nature? And again, I will try and ask you to remember this first. So this is what I have told you, that is what Shilpi's lab was doing. What we intervened is that we measured the mouse brain stiffness, find that 500 Pascal, and we said, okay, we will culture these cells on gel of this 500 Pascal rigidity. So we would like to see what will happen. So essentially what we have here is that parent cells on plastic, recurrent cells on plastic, parent cells on gel, recurrent cells on gel. So we will compare with these four quadrants. First, the migration. Black is for my parent, red is for the relapse. You check if the cells are on the plastic, the velocities are the same. There is actually no difference. Even on a stiffer gel, the velocities are very same. But as soon as you put them on a gel of a soft brain mimicking stiffness, you can see a significant velocity difference here. Then we checked for the invasion, the regular invasion assay, where you put the cells on a top chamber, you have your extracellular matrix here, you give a chemoattractant in this magenta side, and the cells from this blue side invade through to this side, and we image from here. So essentially, if I see more cells, meaning more cells have invaded through. So first, plastic, parent and relapse. You can see relapse has a little bit of more cell number invaded compared to parents, but not really very high. Then we look at parent cultured on plastic versus cultured of bell-like stiffness. Even for the parent cells, when you culture them on the bell-like stiffness, rate of invasion goes up. And then comes for the relapse cells, we really did not expect this. It was like a huge difference in how they invade, right? And that is what you can clearly see here as well from the graph. Further, we looked into the proliferation, what will happen in the proliferation. Again, you can see that the relapse cells and the parent cells cultured on plastic, cultured on stiff gel, not much difference. As soon as you culture them on brain mimicking stiffness, almost a 3 to 4 times difference in the how quickly they divide and they proliferate. Finally, the drug testing. I will just ask you to focus on maybe this part. So we are using temozolomide. If you look carefully for the, again, plastic, black, parent, red, this is y-axis is percentage cell survival. If you give them 50 micromolar temozolomide, difference is not significant. Meaning, both are equally sensitive to the drug. you put the recurrent cells and the parent cells cultured on plastic and gel, and you can clearly see that the relapsed cells cultured on gel, this is red dotted bar, are more cell survival, meaning they are showing more drug resistance. So essentially, we captured these various different phenotypes of the aggressive nature. Now, why this very high innovation? When we looked at the RT-PCR data, what we see here is that to invade what they need to do, because it is a very cross-linked polymer. So first they need to break it. For that they need the MMPs, and the MMP secretion goes up here. So they essentially chop and create the gap to migrate or invade. But to invade, they also need the extracellular matrix to anchor on, and for that they secrete collagen, and that we can kind of see here. So it is a kind of opposing behavior. One hand they are chopping off all the extracellular matrix to create the space, but on the other hand they are also making the road so that they can walk on. And this I will kind of ask you to connect to what I showed earlier, that in the tumor grade 2 the stiffness goes up, probably that time they secrete more collagen, and then as the grade progresses it further goes down because they now try to secrete more MMPs. Finally, obviously the final test is when you put them on the mouse brain. So again you can see here plastic parent, plastic relapse. Compared to parent, yes, relapse is more. When you give them on the brain-like stiffness, both of the tumorigenesis goes up, whether it is for parent or relapse, but for relapse it is much higher. So essentially what we have seen so far, that instead of culturing the cells on plastic, if you bring the mechanobiology aspect, that is you culture the cells on brain-like stiffness, you can capture the aggressive phenotype that is known in the clinical setting in your in vitro studies, and this is what we have seen for all of these: proliferation, migration, invasion, drug resistance, etc. Plastic parent is less than plastic relapse, is less than soft parent and the soft relapse. That is what we have seen here so far. So finally, we would like to check the transcriptomic data between, again, remember those 4 quadrants: plastic parent, plastic relapse, gel parent, gel relapse. We found these 21 genes are differentially expressed in 0.5 kPa relapse, and out of that PLECA7 showed the highest fold change, 4.3. PLECA7 is an interesting protein. It is an adherent junction protein, which is kind of work for the cell-cell junction. But very interestingly, in case of ovarian cancer, the PLECA7 level goes down, but in case of the colorectal cancer, PLECA7 level goes up. So there is no obvious answer that the PLECA7 should go up or go down in a particular context. So we checked that, okay, the PLECA7 goes high, so obviously we downregulated that with PLECA7 siRNA, and as you can see here, I am not going into details, but with the PLECA7 downregulation, the migration invasion goes down, as well as the cells become drug sensitive, the temozolomide sensitive. So the question comes here is that, is it a novel target? We don't know. Question number 2, can we have a molecule which can target PLECA7? Might be, but will that molecule cross the blood-brain barrier? Again, we don't know. But this looks that this can be a novel target that is specifically for the relapsed tumor and not for the parent tumor. So the more details can be found in this paper if anyone is interested to look at. It has far more details. So then Arpita joined my lab, as I was saying. So she did her postdoc from IGIB. She wanted to take this project further, but she is an RNA biologist. She wanted to look into this difference only, the parent plastic versus 0.5 kPa. not the relapse. So she wanted to look at this difference. So Arpita's training is something called LNC RNA. I had no idea, no clue what it was till the Arpita joined. So the LNC RNAs are, as we know that the central dogma say DNA to RNA to protein. So these are the protein coding RNA. But out of all the RNAs, 98% are non-coding. They don't code for the protein. Only 2% of the RNA that code for the protein. So earlier it was thought these are the junk RNAs or junk DNA. But we kind of started to realize no, that is not true. Even if they don't code for the protein, they have lot of the other things to do. One of them is called the LNC RNA, long non-coding RNA. And they have these various roles to play to bring two proteins together, create scaffold, etc. And this has been shown in various different contexts of the cancer that these different LNC RNAs that has been identified in last 10-15 years, which goes up. So now LNC RNAs are actually used as the biomarker for cancers. So in that context, we looked into the LNC RNA NIT1. When we looked at the literature, so low-grade glioma and the high-grade glioma, we see that the NIT level goes up. Normal versus glioma, NIT expression goes up. This is again the relative expression of NIT1. This is the normal. This is various different cell types. In all the NIT levels are up. And again, this is from the TCGA data from the the pool, cancer real patient pool. You can clearly see that with the high NIT, survival rate goes down. So NIT is clearly playing a role in the glioblastoma context. Does it play a role in the context of the mechanobiology? That was absolutely untouched, and that is what we tried to look into. So here comes my collaborator, Dr. Mohit Jolly's, help. That we looked into the TCGA data, we looked into the whole bioinformatics, and what we have found here is that in various different cancer-related pathways, in case of G.B.M, there is a positive correlation with the NIT1 as well as those pathways getting upregulated. Not true for all other cancers. There is a mix, but here everything kind of goes up. So we thought, okay, we'll go ahead with the NIT1. Further, we also looked at the various different pathways which are related to the mechanosignaling. Again, Rajdeep was talking about these pathways yesterday. There also we see a very nice positive correlation. So we thought that the NIT1, cancer and the mechanobiology may have a role to play together. So again, what Arpita did was that plastic gel glioblastoma cells, and you can see that again here I am not talking about the relapse cells, I'm just the parent cells. The NIT1 level goes up when you culture them on soft gel. Good. Next we look at various other invasion related markers. So these are all RT-PCR data. Invasion related marker MMP2, MMP9, fibronectin all goes up. Staininess related marker OCT4, SOX2, nano, blah blah all goes up. Epithelial to mesenchymal transition, epithelial marker goes down, mesenchymal marker goes up. So looks at that when we cultured them on the soft gel, the NIT level goes up along with many other things goes up or down. Is it just a correlation or the causal? So the obvious next thing to do is to downregulate NIT1 with siRNA to bring it down to the level of plastic that we did, and we see that we can rescue all of the phenotypes, which kind of tells us that soft substrate hits on NIT level goes up, and that make all of these changes, not just at the RT-PCR level. We have also checked at the functional level, migration, invasion, you can see the changes here. So, how it is working? Now, when we talk about the mechanobiology, the sensing of the stiffness, that always happens through something called actomyosin contractility. that cells adhere to their surrounding, attach to the focal adhesion, and using the myosin motors they pull to measure the stiffness. So the actomyosin contractility are generally at the center of all of this mechanobiology sensing. So what we did with any mechanobiologist will do that we use LADB, active inhibitor, blebistatin, myosin inhibitor, LPA, which increases the actomyosin contractility. And what you see here, I will just ask you to focus on these two. On plastic, where the contractility is high, and the knit level was down, I block actomyosin contractility with blebistatin, knit level goes up. On soft substrate, where the knit level was up and actomyosin contractility is down, with LPA, when I increase the actomyosin contractility, knit level goes down. So it is the actomyosin contractility and knit, they have a kind of opposing role here. Further in the literature, I have already told you one of the stemness gene, SOX2. We also see that the NIT1 and the SOX2 are correlated, at least we got from the literature. We also checked in our case and we found yes, in the various different conditions, wherever NIT level goes up, SOX2 level also goes up. So probably NIT is working, stiffness, NIT working via stemness gene SOX2. One thing when we were doing it, we used mepodazole, which is a microtubule targeting reagent, which target the microtubules. What we found is that the microtubule targeting reagent, and as you know, many chemotherapeutic drugs are essentially microtubule targeting reagent. Both NIT1 and SOX2 expression goes very, very down. That tells us that okay, some microtubule targeting reagent should be a novel drug to use. However, mepodazole or other MTA drugs are not good for the glioblastoma because that does not cross the blood-brain barrier. We looked into all the drug trials that are going on, and what we found was that there is one drug, varibolin, which is now in the phase 2 trial for the G.B.M, and which is a microtubule targeting drug. So we thought that we can try the varibolin, whether this will work on NIT1 and can bring the NIT1 down. So essentially, will the varibolin bring all of this down and make the cells more drugs, TMZ, temozolomide sensitive? Yes, so when we did, we see that yes, temozolomide IC50 value on plastic is here. When we do it on gel, it goes up. That is meaning the cells are not sensitive to the drug. But when you use the varibolin, that is here, you can see the cells become again sensitive to the drug and the IC50 value goes down. So that kind of tells us is that varibolin with temozolomide in combination actually might work well. And because it crosses the blood-brain barrier, it is already established. So this is the model that we kind of think that it should work. So how much time do we have?
We have almost 30 minutes.
30 minutes. Okay, good. So basically the details, again you can find these preprint. This is soft versus stiff. Erpita is also working with 2D versus 3D, which is here. I am not going into all the details, but the take home message here is that whatever we have seen, soft versus stiff, that is on soft, NIT1 level goes up, cells become more aggressive, SOX2 level goes up, blah blah blah. Everything happened when you go from 2D to 3D spheroid. Again, yesterday a lot of people talked about spheroid, but at a much kind of more prominent way than we have seen in case of soft versus stiff. So these also are available on the bioarchive. We are now communicating the paper. So here I will kind of shift the gear. So I have another 10 minutes. So far I was talking about the science. I was wearing my scientist hat. Now I will wear my engineer's hat and I will change the gear a bit. So as I said, one part of our lab is working on the mechanobiology. Other part is working on the microfluidics and microfabrication device, etc. So these are the few examples that we are kind of working on. Multi-drug testing, spheroid culture, some barrier on chip. In fact, that's why I was asking about. So we are working on placenta on chip, and we are looking what can cross the placental barrier. And here you can see that the dynamic deformation on chip, what we are trying to make for the atherosclerosis model. So that is what that in the dynamic stenosis what will happen. So that is something that we are working on. But I will just talk about these two for today in next 10 minutes. So the first one, that is this multi-drug testing tool. So when you test the drug right now, what we generally use is the 384 well plate, create the different concentration manually, lot of work, prone to manual error. And if you have to do the multiple drugs, things become even more complicated. Solutions here are either the robotic arms, which are very, very expensive, or the microfluidic chips, which has a learning gradient, learning curve. Not every lab can adapt it very easily. So we thought that can we make something that is easy to use, almost off the shelf. One will take it from the shelf, use it without any training. So this work is done in collaboration with Professor Prasanna Gandhi from Mechanical Engineering and two of my graduated students, Ketki and Sheetal. Now both are in industry, one in USA, another in India. So this is what we have made. So this is essentially the lot of the microchannels going in perpendicular with one another. You can say these are the two reservoirs. If you add the drug, you can imagine these colors are the drug, colors of Indian flag. They will create a nice gradient here on this channel. So essentially all of these points will experience different concentration of the drug. Beauty here is that the shape of this channel is in such a way that wherever these two channels meet, that we call as a node, there we have a little dome there. What that dome does, that we introduce the cell from this inlet. When the cells pass through with the flow, as soon as they come to the dome, because of the increase in the cross-sectional area, flow velocity goes down. And when the flow velocity goes down, like a river, if the velocity goes down, it will deposit sands. In these nodes, cells get specifically deposited. And that is what we get, that in all of these nodes, we get the cells get deposited. Obvious question comes, do we get a uniform distribution of the cells? Yes, we get that. Obviously, after a lot of optimization in the dimensions and other things. Next question. Will this gradient be stable? Using the fluorescent, FITC we have checked, and yes, the gradient remains stable, at least for the time scale for which most of the drug testings are done. So what we do here? Essentially, we have one reservoir, one source. In the first you put the cells will get nicely distributed everywhere. Then in the source well, you add your drug, you get a nice gradient of the drug, you expose the cells to, and this thing size is that it will fit on 1/2 of a slide. So 1/2 you can have this, in another half you can have your control. So in one case, the cells in each of these nodes will experience a gradient of drug. In control, they will I will not experience any drug. And that is here the control and the test. After the test, you just simply stain the nucleus, put them under the microscope. I don't know if you can see from there, but here where the concentration is high, less nuclei, because most of the cells have died. As you go here, the number of nuclei goes up. In the control, it is remaining constant. So essentially, you can take the ratio, which will give you that the percentage inhibition in each of the nodes. And from our simulation, I know each of the node what is the concentration. From that, I can generate this data, which will tell you what is the IC50 value or the 50% inhibitory concentration. Beautiful part is that because it is made of PDMS, after your experiment is done, you can fix the cells. So in each of these 100 spots, cells will get fixed which have experienced different drugs. You can take the PDMS off and put the whole chip for immunostaining so that you can also see how the drug works. Here we have used curcumin, which is a microtubule targeting drug. You can see where the concentration was less, microtubules are intact. Where the concentration was high, microtubules got disrupted. Further, you can use the drug combinations in the various different ways, and that is what we kind of show here. So you can also comment on how the different drugs will work. I think I'll just skip this part in the interest of time, but we also kind of work on this spheroid making device. Yesterday, Pallav was talking about the spheroid making device, easy to make. We are also trying to do something that any lab can make without any available instrument, even the lasers. So what we did is that we take a 384 oil plate. If you take it upside down, you will see that the bottom side is like a nice oils. So we use that one, transferred the structure on a gel, shrunk the gel, reduce the size, transfer it to PDMS, this multiple size reduction from this size of the well that get reduced to this size, which can fit into a, this got completely blurred, which can fit into a nice 6 well or a 12 well plate, where we can do a drug testing on the spheroids. And I have already showed you in the glioblastoma is that 2D versus 3D makes a huge difference. I'm very sorry that it got completely blurred. Okay, you have to now trust me that also we can create uniform size distribution as well as a gradient size distribution. This is a gradient size distribution which got blurred, but you can kind of see here. So if you want to know the effect of the drug on the size, that you can do. We can also do the co-culture. So the beauty here is that you have 100 spheroids, each spheroid of a co-culture of 2 cells of different ratios. So if you want to know how the 2 different cell types in different ratios will work, against a drug, that you can kind of use this device to work on. So this was my last slide, and one more. That is, you can also connect this device with the syringe pump essentially. So what it takes is that you take the device, you put your cells, you switch on the syringe pump, and you go home. Come after 5 days, your 600 spheroids are ready from a 6 well plate without investing any sophisticated instrument. So essentially, what our take-home message here is that probably I could have convinced you that in the context of the cancer biology or other, even the cell biology, mechanobiology plays an important role, and considering the tissue microenvironment and the tissue mechanics is crucial. And the mimicking can come from the microfluidics, from the mechanobiology, from the various aspects. And this, again coming back to the aim, microfluidics, materials and the mechanobiology together can unravel some very, very interesting biology. So with that, let me thank, everything is blurred. So let me thank my group members. As we always say, they do all the hard work. We just present here and enjoy the nice weather in Paris. So the recurrent G.B.M story, this was the group in collaboration with Shilpi. In CRNA G.B.M story, this is the group in collaboration with Professor Mohit Chali from IISc. Concentration gradient this is the group in collaboration with Professor Prasanna Gandhi. And the spheroid story, this is the group. And as I say, no work can be done without money. So obviously, thanks to all the various funding agencies. And funding agencies don't mint money. So essentially, the money comes from the citizens of the country. Thank you.
Long live India and France friendship.